where do sirtuins enter the story here? Because one of the prevailing ideas that I've heard is that Na depletion is affecting aging within the cell, potentially some of these repair mechanisms through modulating sirtuins.
Sirtuins. Wow. So yeah, I told you that NAD coenzymes are the central catalyst of metabolism. I told you that one of the first results that we got was that in our extended lifespan in yeast, that depended upon a gene called Sir2, right? So there's actually two models of aging in yeast, and one of them depends on Sir2. The other one is actually opposed by Sir2. The one that depends on Sir2 was popularized by Guarente and Sinclair and others. And it's basically the number of times that a yeast mother cell can divide.
You've explained this to me a number of times, and it's a bit to get your head around.
Is it hard? So it basically, if you imagine that there's yeast growing in a flask, right? Because we're trying to make beer, wort, or wine. So we have a flask of yeast, right? And then, we're going to get back to that flask, but let's flame a metal loop, okay? Sterilize a metal loop, just like Louis Pasteur, and we'll dip it in there and we'll streak it out, and then we'll take single cells. We'll take single cells and array them on a petri dish, right? And then every time that single cell gives rise to what's called a daughter cell, we're going to throw the daughter cell in the trash can.
So you're only retaining the mothers.
Right. You're only retaining the original cell that you put on this dish. And then you install a graduate student or a postdoc in front of a dissecting microscope for a week or two, okay? And every 90 minutes, the mother cells produce a daughter cell. The graduate student throws the daughter cell in the trash until the mother cell has divided 20 or 21 times.
What's the rationale for that? Why would the graduate student be told to do that?
Because what they're trying to find out is how many times the mother can produce a daughter. How many times the mother can produce a daughter? You can answer that question. Okay, the answer is 21 times or something on average. And if you calorie restrict them, if you put lower level of glucose on the plate, the mother can do that more times. Okay, so you can measure that result. The problem is that if you go back to that flask and you try to look for mothers that have divided 21 times, they would have literally 21 bud scars on them. You can do the math on the frequency of those cells in the population. Would you like to know what the frequency is?
You've told me before.
Yeah, 1 in 5 million. So the phenotype, the aspect of living, we call it a phenotype. The phenotype that this thing, even presents itself in, is a phenotype that only 1 out of 5 million cells ever experiences. Only 1 out of 5 million cells can ever be. You'd have to have 5 million cells in a culture for there to be one cell like that.
Connect the dots here. So why is that important? That model, but what you're saying is that model has then incorrectly been used to study longevity?
Yeah, it's actually mind-boggling. So I really love yeast. I spent a lot of my professional life working with yeast. I purified a prohormone processing enzyme from yeast. So the enzyme that we have in our body that makes insulin and proenkephalin is pretty much the same human enzyme as a yeast enzyme that I purified in graduate school. So I have nothing against yeast, but yeast is not a good model of aging, especially that model is not a good model of aging because that's not a selected trait.
So how did studying that model, in your view, from your perspective, lead to incorrect conclusions about sirtuins?
So Sirtu is important in that model, right? And
when you say it's important in that model, that 1 in 5 million circumstances, sirtuins acts as a longevity gene.
I installed my graduate student Peter Belenke in front of the dissecting microscope in 2007, and he showed that NR, increased Sirtu activity, and it gave yeast a longer lifespan. So they had a greater replicative lifespan when they had NR than when they didn't. But the reason that we didn't claim that is suddenly, makes NR a longevity drug is that's yeast, not human, and that's an incredibly rare yeast cell.
So does Sirtu affect longevity in all of the other yeast cells?
In the worst possible way, ironically. So if we go back to this flask that we have of yeast, because we're going to, let's say, inoculate it into some grape juice to make some beautiful Australian wine or California wine. If we let's say we have a lab, outside the vineyard, right? And we have grown these yeast cultures in it's now May. Let's say we've grown these yeast cultures in January, February, March, April, and May. So we have 5 cultures, right? And then today we go into that lab and we plate the yeast for viability, right? We see what percent of the cells are still alive. It turns out if you delete the Sirtu gene, you get better chronological aging. You get cells that can live for months or years as opposed to days or weeks.
So if that is so clear in the research, my understanding is that the research from those mother yeast cells led to a plethora of ongoing research into sirtuins, companies set up, etc.
Yeah.
So how
this happened?
How does that happen?
If it is claimed as a pro-aging gene from yeast, right? And I'm pointing out that it's 1 out of 5 million cells, and then Walter Longo, who's a very well-known molecular gerontologist, molecular aging researcher at University of Southern California, here in Los Angeles, showed that deletion of Sirtu is better for chronological aging. But his paper was not until 2005.
Is that the same in humans as well? Deletion of Sirtu would lead to longevity?
No, Sirtu doesn't have anything to do with animal longevity. The problem is that in the early 2000s, there were incorrect, non-reproducible papers published in Worms and Flies that claimed that extra copies of the worm and fly Sirtu related genes extended lifespan. But the problem is that that's not a reproducible result. So the claim was that extra copies of the Sirtu gene extend worm lifespan, that extra copies of the Drosophila Sirtu gene extend fly lifespan. But when independent research groups tried to reproduce those results, they could not reproduce those results. And in fact, when you delete one copy of the fly Sirtu homologous gene, you get a fly that has extended lifespan when it's amino acid restricted. So the Sirtuin genes are not candidate longevity genes in animals. They were declared to be anti-aging genes in animals on the basis of research that has no value. In fact, the only value it had was to churn money on, poorly interpreted and overhyped work.
This episode is proudly brought to you by Inside Tracker. Track your blood biomarkers, understand your biological age, and receive personalized lifestyle tips backed by evidence to optimize your health. To get started with Inside Tracker today and get 20% off your first purchase, head to insidetracker.com/Simon. That's insidetracker.com/Simon for 20% off. In one of your reviews, this is a quote, you said, "The asymmetries between publication ease and rewards of positive versus negative data have been magnified by commercial and reputational interests that are deeply tied to tested and disproven theories." What does the kind of negative data have to do with this story?
Yeah. So let's go back to the late 1990s, right? There's a very nice paper by Guarente and Sinclair. I like two author papers. Like some of the best papers from my lab have been two author papers. One, trainee and the boss. Okay. So there's a classic paper of Sinclair and Guarente, 1997. Is a true paper. Okay. The conclusion was that David Sinclair showed.
When you say it's a true paper, what do you mean by that?
I mean, it's been reproduced by other people. The insight that came out of that paper is evidence-based. Okay. The problem is it's also been massively overinterpreted. Okay. But the initial result was true, which was that if you go back to that mother-daughter thing where the phenotype occurs after 21 generations, the mother cell is retaining what are called ribosomal DNA circles, which you've never heard of because humans don't have ribosomal DNA circles. Yeast has ribosomal RNA encoding genes that encode ribosomal RNA, or called ribosomal DNA, that are repeated one after the other. Because you need a lot of ribosomes, so you have, if you're a yeast. And so the ribosomal genes are arrayed one after the other. And then David found that those things pop out into a circle, and the mother cell fills up with these ribosomal DNA circles. And the daughter cell is like a new cell because the mom kept all the circles. David found that process of controlling the ribosomal DNA circles was dependent upon the gene called Sirtu. That's a fact. That's true. And no doubt when my student, Peter Belenke, showed that NR extended yeast lifespan, it's helping those yeast mother cells control the number of ribosomal DNA circles. Okay, so that's a result from 1997 that's true. It helped us do something in 2007. Here's where it's not helpful. Expecting that activity in an extremely rare yeast cell had some fundamental thing to do with aging. That was simply storytelling. It was storytelling by Sinclair and Guarente and a lot of other people.
Was that for the purpose, do you think, and I know you would have to speculate, but for getting more funding to do further research?
Well, we work hard in the laboratory. And so when you get a result, you want to maybe spend some time speculating of what the larger implications could be. Okay? But testing the idea that that a yeast cell would have anticipated the causes of death in an animal is a pretty big stretch. But they're entitled to test that. The problem is that when they got positive results, they went straight to press with them. And they claimed that the Sirtu gene that they found, actually, they didn't discover the Sirtu gene in yeast. It was a known gene called silent information regulator 2. But they found this, aging phenotype of Sirtu in yeast. And then they tested it in worms and flies, and they published positive results that were wrong. Now, the problem is that someone else can't reproduce that and has a negative result. Well, it's very difficult to publish a negative result because, the supervisor can say, well, maybe you didn't do it right. Maybe you're not as talented as the Guarente lab. You didn't get this positive result. Or the journal editor says, well, that's not exciting. We're interested in exciting results. And so it might go to a sort of archival journal. It might not be put into a paper at all. It might just be left in laboratory notebooks. All of those things are true in the Sirtuin field. There are things that were never published because they couldn't be reproduced. People spent a lot of time and treasure to try to reproduce these results. They couldn't publish them because they couldn't get any positive results. In 2011, fully 10 years after reports started appearing in the literature that worm and fly Sirtuin, we call them now Sirtuin genes, Sirtu related. 10 years after the initial reports that animal Sirtuin genes are anti-aging genes, a group of investigators that included David Gems, Linda Partridge, the best aging molecular biologists in the world, were able to publish a paper in Nature saying fly and worm Sirtu overexpression does not extend lifespan. But that was very rare to be able to publish in a high-impact journal like Nature a negative result. Yet even that didn't derail the Sirtuin story. The people, the partisans involved in this continued to say that Sirtuins are longevity genes. They continued to say that they had activators of these genes. Resveratrol was one of the proposed activators. And nothing stopped this, to the point that, there was a biotech company called Sirtuins, and I know this very well because they were interested in my intellectual property. They had me in as an advisor. I was present at Sirtuins meetings. I saw the low quality of data being, examined at Sirtuins. They were looking at resveratrol as a possible SIRT1 activator. They were considering SIRT1, human SIRT1, as a human longevity gene. And they basically sold this company to GSK for $720 million. And as you know, Sirtuins are a failed, drug target. Why? Because they're not longevity genes. There are no activators of these genes. And the whole hypothesis was nothing more than...
There are no activators of these genes.
Resveratrol is not an activator of these genes. Higher NAD increases the activity of all NAD-dependent processes.
If David was sitting here now, how do you think he would kind of argue his position or his view to counter what you're saying? If you were to kind of steelman his position?
I don't know, because when I have challenged him on all of these things, he's basically blocked, the discussion. So I'm blocked on Twitter. I can't see what he, says. People send it to me. And then when it's, quite egregious, I comment on it, but he doesn't directly respond to my critiques. He repeats things over and over again that he found, he and others in the Guarente lab found Sirtuins as longevity genes in yeast. He doesn't go into the details of whether it's affecting 1 in 5 million cells or the whole culture. He doesn't deal with that at all. He's maybe never cited that work. He still claims that resveratrol is an activator of SIRT1.
I think I did read one of his papers, and he did mention that it was in that replicative model, at least.
But he won't point out that the phenotype in the replicative model is a phenotype of 1 out of 5 million cells, and it's a dispensable cell. He doesn't deal with the idea that the targets of Sirtu are not conserved. He thinks that the worm and fly results are replicable when it's known that they're non-replicable. He acknowledges that there were early papers showing that resveratrol was a biochemical artifact where it was interacting with something called a fluorogenic reporter group. But then he's come around, and at very great expense, he's developed a theory that resveratrol binds to two particular amino acids in human Sirt1. The problem is that those amino acids in human Sirt1 are not in yeast Sirtu. And he claims to have discovered resveratrol using yeast Sirtu. But resveratrol doesn't extend lifespan in yeast, even in that model. And the ability of resveratrol to activate the biochemical activity is known to depend upon this fluorogenic reporter group. So David's stuff has been falsified.
So do you think resveratrol, fisetin, pterostilbene, these kind of, I guess, similar compounds that people have claimed are activating sirtuins, do you think they have any benefit at all through other mechanisms?
There's no use case for resveratrol. It's not, bioavailable to humans. And there's been enormous treasure, put into trying to find some beneficial activity of it.
Does it matter if you're having it with fat, the bioavailability?
It might, but there are not convincing human data showing benefits. Pterostilbene is a more bioavailable compound than resveratrol, but there are human data showing that pterostilbene elevates low-density lipoprotein cholesterol in human beings. And so, in fact, the company that I consult for, Chromadex, was originally a source of a particular formulation of pterostilbene. We were selling it to a company called Elysium, along with nicotinamide riboside, for them to put those two compounds together in a supplement. They literally had my patent numbers on the bottle of Elysium basis when they initially developed this compound. They had NR, where the IP came from me and Dartmouth, and they had pterostilbene, which is something else that was developed by Chromadex. When I looked at the human data that were coming out on pterostilbene use, it became clear that this is a compound that in a dose-dependent and time-dependent manner is elevating LDL cholesterol, bad cholesterol, right, associated with atherosclerosis and a lot of bad human health outcomes. And, I went to the leadership at Chromadex and I said, if we're in the, wellness business, we shouldn't be selling this stuff. We stopped selling it. And so I don't think there's a use case for pterostilbene.
So if there's not a use case for these compounds that have been labeled as sirtuin activators, and as you've spoken to, don't seem to even be activating sirtuins. You've spoken to the fact that in 2011, I believe you said that paper came out saying sirtuins are not longevity genes.
In worms and flies.
In worms and flies.
And they're also not in mice.
So where's the field at today in the rest of the Guarente lab? Is that a consensus? Would you say most people are in agreement that sirtuins are not a key part of human longevity?
I think it's only highly partisan people in the scientific community that would argue that they are, longevity genes. I don't really see anyone active other than David Sinclair defending the idea that sirtuins are longevity genes. I haven't heard much from Lenny Guarente on this subject in a while. I spoke extensively to Shin at a conference last summer. He said that he doesn't really think that he doesn't believe in the concept of monogenic longevity genes. So I think that David is kind of out on his own on this one.
Do you think you could have a conversation with him?
Oh,
or is it too personal?
Yeah. No, I would love to. Let's hereby invite him. I'll scooch over.
Well, I'm an Australian, so it might make it easier.
Okay. Well, I know that he spends time, here in Los Angeles, and I would love to have a conversation with him. We could find out what his argument is in favor of Sirtuins, and we could talk about his ideas about the the information theory of aging as well.